back, everyone, to attempt 2 of the tomography lecture in our course. So I'm very happy John will do that today.
So I'm going to talk about tomography and subtomography averaging. I should start by apologizing for anybody that showed up a couple of weeks ago. I was in no fit state to give a lecture. So I'm going to start with the question, what do we need to get a 3D structure? We need sample preparation methods. We have to be able to get our sample ready in a manner that we can go on and image it. We need a microscope. We need different views of our object of interest. So we need to see it from different directions. We need computational approaches for producing a 3D reconstruction from those 2D projections, and we need methods for validating and interpreting the 3D structure. This goes for single particle reconstruction as well, of course. And the difference, or the special thing about tomography, is how we get the different views of our object of interest. So this is a projection image of a hand holding a pen. If we were doing single particle reconstruction, I'd get all of you to put your hands and your pens in the air, and we'd take a projection image of you, and we'd reconstruct hand and pen from all of your hands. In tomography, we're going to physically rotate our hand and our pen within the microscope and image it from different directions. That's important. We tend to over-interpret 2D projections. I mean, you see this as a hand holding a pen, but you actually don't know whether the person is holding the pen this way, or whether the person is holding the pen this way, or whether that's a surgical insertion between the thumb and the finger. But if you rotate, you obviously see that directly. So that's the difference. In single particle methods, we obtain different views by imaging many different copies that are oriented differently relative to the beam, and in tomography, we obtain different views by physically rotating the sample in the microscope. I'm going to show a short movie from the Baumeister department, which illustrates the process of tomography. Here are, in this case, virus particles that are happily floating around in some kind of buffer solution until they're dropped into liquid ethane and freeze instantaneously, effectively. And then we have so we end up with a thin layer of sample, which is on an EM grid. You can see this is a side entry holder. It's stuck into a microscope. Rotated, it's imaged, an image is collected onto a camera, and the holder is rotated, imaged, and we do this repeatedly. And in this way, generate a whole series of different projection views of this, in this case, what is a little cluster of virus particles. There are many more images collected than this, and these are then reconstructed using back projection to generate a 3-dimensional volume, which shows that set of virus particles. As in single particle, this is a 3-dimensional reconstruction. We can slice it in different directions, and very often, especially in tomography studies, you see isosurface renderings with different components colored differently. In this case, the outside of the virus showing the glycoproteins, and the inside showing the core. If you look at this image, this illustrates 2 of the things we're going to look at today. The outside of this virus is very, very noisy. That is direct interpretation of the tomogram. The inside core looks much clearer. That has been averaged. So there's a subtomogram averaging step that's gone on to produce that core reconstruction. So the data as it comes off the microscope is what we call the tilt series, and it's then reconstructed to generate a tomogram. So this is a tilt series. So this is just rocking between minus, whatever it is, probably minus 50 or minus 60. You can see, in this case, these are an in vitro virus-like particle. You can see black gold fiducial markers. These are 10 nanometer gold beads that are used for the reconstruction. And you can see that as we go to higher tilt angles, the image gets darker, and that's because at higher tilt angles, that slab of density appears thicker to the electron beam, and less electrons get through, so we end up with a darker image. So that's the tilt series, that's the data, and from that we generate a tomogram, and now this is being viewed as slices going up and down through the density. So we're now going from the bottom up to the top, and in this image you can see that these virus particles are hollow. You can see a hexagonal lattice if you look closely on the top and the bottom of the virus, and that's then the tomographic reconstruction. So why do we want to do tomography? There are I think basically two main reasons. The first one is if our sample is a unique structure. If the thing we want to look at in 3 dimensions is a bit of a cell, a bit of a tissue, then we can't average many copies of that together because that's a unique object. So if we want a 3D reconstruction, we have to do tomography. The other reason may be because our sample of interest is within some complex environment. And here I'm talking about what might be a protein complex, something where we can average. But if you imagine a thick layer of ice and we have our protein complex in the middle of it and many, many other protein complexes above and below, then in a single 2D projection, as you have a single particle, you're not able to align that particle due to all of the noise effectively that's contributed from the particles above and below. But if we do tomography, we may be able to then extract that complex and align and average it by subtomogram averaging. So I'm going to talk at least briefly about all of these things, about sample preparation methods, about data collection and microscope requirements, about alignment and reconstruction, and about subtomogram averaging. So let's start with sample preparation. So what are the requirements? The sample must have the structure of interest preserved. That sounds like a trivial thing to say, but the vast majority of tomography is still done on samples which have been put in resin and stained. And they will have certain things preserved that you can then interpret, and other things may not be properly preserved, and therefore you can't interpret them. So the thing which we're interested in has to be preserved in the sample. The sample needs to be thin enough to generate projection images, and the sample needs to be stable in a vacuum. There's a whole zoo of methods for preparing samples to do cellular EM, if you're interested in the structure of a cell. These can be loosely grouped into methods that involve chemical fixation as a starting point, and methods that involve cryofixation as a starting point. And these methods, the most common methods involve ending up with a sample in resin, which you can then section using a microtome to get thin layers, which you put in the microscope. Even if we use cryofixation, we may then go into resin in order to do sectioning, staining, and again, room temperature electron microscopy. But the things which I'm going to talk about today are what one might call the true cryo methods. So these are methods where the sample is frozen by plunge freezing or high pressure freezing. So it's vitrified, and it's not then embedded in anything else before it goes in the microscope. So that vitrified sample is either sectioned with a knife, that's Semmelweis, or is cut by Fib milling before it goes into the microscope. One thing this is for cells. Of course, if we're looking at something like virus particles, we may be able to simply plunge freeze and then go directly into the microscope. So first of all, Semmelweis. This is the equipment that's available here at the LMB. So we have some kind of high pressure freezer. This allows you to vitrify thicker samples than you can vitrify by plunge freezing. And it works by placing the sample under very high pressure at the point where the temperature is dropped. And as you all know, ice has a lower density than water, so it requires some kind of expansion. So that forces you into vitrified ice states. And then this is a cryo microtome, which is what's used to then cut that block of sample into thin slices for putting in the microscope. This is a slide from Peter Klander. This is a zoom in on a cryo microtome. You can see the grid here, zoomed in here. The sample is on here. And then there's a knife, which you can see here. This is on the zoom in. And the sample is then drawn over the knife, and then repeatedly sections are cut, and they end up joined together and form this ribbon that's pulled over with an eyelash onto the grid. This is then an image through a sample that's been prepared by Semmelweis. And you can see very nicely cellular features, but you can also see that the sample is compressed in this direction. And these are this is one of the artifacts that are associated with the fact that we physically cut this block of vitrified sample with a microtome. The other popular method for thinning samples is Fib Sem, and this again is the equipment that's available at the LMB. So this is currently most commonly done with cells that have been grown on electron microscopy grids and plunge frozen, but the same approach in principle could be applied to high pressure frozen samples. And in this case, if we imagine this is a cell on a grid, we identify a region which we want to preserve. This is this green slab. And then if you imagine we're now looking onto that cell in the direction in which we're going to apply the ion beam. So 2 squares are marked, and this region are then exposed to the ion beam. So if we imagine region 1, this then removes this part of the cell, and then region 2 then removes this part of the cell, and one is left with this very thin lamella which is suspended between the two sides of the cell, and this region is then thin enough to image by tomography. So this is what this looks like after removing the top and the bottom and then rotating so we look down. It's a beautiful example from the biomolecular department. This is a view of a lamella, and you can see ribosomes and microtubules and all kinds of details. These samples don't have the compression artifacts associated with Semovis, but this is obviously a complex and challenging method to apply. So what about data collection? So we collect an image. Let's imagine we've collected our first image, we've put our sample in and collected an image. We're then going to rotate the stage, physically rotate the stage, to set a new tilt angle. Stages are not mechanically perfect, and we're potentially imaging at very high magnification, and that means when we rotate the sample, it may move out of the field of view, or it may shift within the field of view. We therefore have some kind of tracking region. So this is another region of the grid which we're imaging. So we track, we compare that image to the previous one to work out what movement has taken place, and we correct for that by shifting the stage, or in some case by beam image shift. We'll then focus the sample using some other area to do the focusing, and then we'll collect an exposure. Then we'll rotate the sample again to set a new tilt angle, track, focus, and collect an image. That's the kind of standard process. Depending on how your microscope performs, you may not need to focus at every step, you may not need to track at every step. This obviously requires that you're analyzing the images that are coming out of the microscope as you're collecting the data. And if you imagine we might do this 40 times in order to collect a tilt series, each tilt series then takes quite a long time to collect. So we end up with a much smaller data set than we would if we do single particle. In order to be able to do cryo-electron tomography of thicker samples, we need certain microscope requirements. We ideally want a high voltage microscope in order to be able to penetrate a thick sample. We want an energy filter, again, because we're probably looking at a thicker sample. We want a very stable and well-calibrated microscope stage, because the less we have to correct for when we rotate the stage, the more efficiently we can collect our data. And we obviously need appropriate software that allows us to analyze the images as they come off, control the microscope, and collect the data. So we're going to collect a tilt series that looks like this, and we're going to reconstruct a tomogram that looks like this. So what we've done in the microscope is we've done forward projection. We've taken our 3D object and we've generated projection images that are 2D projection images, and we're going to reconstruct this by back projection. So we're going to go from our 2D projections to our 3D reconstruction. Now our holder is a physical object, and as we tilt it, our sample is getting increasingly thick, and at some angle, the holder just gets in the way and you can't tilt any further, and even if your holder didn't get in the way, at some point your sample becomes infinitely thick. Now that means that we can only tilt to a certain angle, which is typically 60, 65 degrees. So if we think about what's happening here in Fourier space, each one of these projection images contributes a slice of the Fourier transform of the object. So each one of our projection images contributes one of these slices in Fourier space, and that means that we have missing information in Fourier space. So we don't have the side views of our object. So we have incomplete data. And that means that our 3D reconstruction doesn't look uniform. So this is an XY slice. So this is the view from above, effectively, but a slice through that. And you can see that our objects look nice and round. If we go back, that's this slice here. But if we look at in the other directions, we can see that what should be a sphere, a round virus particle, is stretched at the top, or the information is very hard to see at the top and the bottom. And that's the effect of the missing information, the missing wedge. We also have missing information due to discrete sampling. So as we go further out, then are gaps between the Fourier planes, because we haven't used an infinitely fine angular step. These planes are thicker for thinner objects, and there is a resolution to which the information is filled in, so after which we then have missing information due to discrete sampling, and this resolution limit is defined by the Crowther criterion. That's only relevant if we're interpreting data in the tomogram to that resolution, which we may not be doing, and we're not doing in the case of subtomogram averaging, so we can come back to that. Okay, so that's what happens. We end up with a reconstruction which has missing information. And as you can imagine, how much information we have and what information is missing depends upon how we collected the data. So if we went to higher tilt, we'll have a smaller missing wedge. If we use a finer angular increment, we'll have less gaps between our planes and so on. So we need to ask ourselves certain questions before we start data collection. Obviously, the way we collect the data influences the data that comes out. And there are lots of questions to ask. So what total dose should we apply? Should within our tomogram we be applying 40 electrons per square angstrom or 140 electrons per square angstrom? The higher the dose we apply, up to a certain point, the more signal to noise we'll have, but the lower the interpretable resolution will be because the higher dose will have damaged features that we'd like to interpret. What tilt range should we choose? So should we be doing plus or minus 50, plus or minus 60? The payoff here is the completeness of information versus the fact that if we collect more images, we may need to put in more dose. And if we collect more images, it takes longer to collect our tilt series, so we get less data in our microscope session. What angular increment should we be collecting? Again, this is the resolution to which our planes overlap versus the total dose again, if we need higher dose in order to collect more images. And again, the speed, fewer images takes less time. So if we're only interested in very low resolution features, and we've got quite a then we may be able to get away with a larger angular increment. What order to collect the images? So it used to be the case that we were rather limited in collecting sequential images in order to have effective tracking. These days we can collect images we can collect the 30 and then the minus 30. We can collect images in almost any order. So we need to decide what order we should collect, and that influences the speed of the data collection, the reliability, the optimal dose distribution, and the distribution of sample distortions, and I'll come back to that question. What magnification should we collect our data at? Obviously, there is some resolution limit set by the pixel size. So if we work at higher magnification, we can go to higher resolution before we're affected by Nyquist. If we use a higher magnification, we have a lower field of view, so we're seeing less interesting things. And there's also the question of the decree of the camera. If we work at very high magnifications and then Fourier crop our data down, then we can make use of the very high camera performance at lower frequencies effectively. And finally, what defocus should we collect our data at, which is a balance between high frequency signal and low frequency contrast effectively. So lots of questions to think about before you start. So once you've got your data, how do we reconstruct a tomogram? This involves alignments and reconstruction. This is comparable to single particle reconstruction. So we have a set of projections of our object. We need to know how those projections relate to each other, the angles and the shifts between the projections. This is alignment in single particle, and it's the same thing in tomography. The difference is that we've defined the angles in the microscope because we've physically tilted the object around the defined axis. And the axis of the microscope is extremely reliably positioned relative to the camera, so we don't necessarily need to solve for that if we know where it is. We do, however, need to do alignments to deal with the shifts in the image. As we rotate, the sample has shifted, and we've corrected for that, and that's not done perfectly. So what you see here is the data as it comes off the microscope and the shift-aligned tilt series. And as you can see here, it's jumping around, and then after that's jumping around, and then after alignment, it's perfectly aligned. This is actually a particularly good example. Those of you that have done tomography will have seen things jump around much more than that. So we've now got our shifts aligned, so we know how all of our images relate to each other, so we now need to generate our reconstruction. And typically in tomography, that's done by weighted back projection. So that's as simple as if we imagine this is now a 2D rather than a 3D example. We have our object, which is 3 dots. We have a projection image collected in this direction, and we're going to back project it out. So that would be what one projection back projected would look like. That would be the result of 3 projections with 40 degrees between them, and so on and so on, and eventually we end up with something that looks like our object, in this case 25 projections with a 5-degree increment. There's another way of explaining the same thing. If we have, but we're doing a 2D version, so we have a disk, we project in this direction, that is the projection, so it's a 1D projection, and we can then back project to generate the volume again. That's the result if we have a single image. In this case, if we have 3 images, we end up with something that looks closer to our object, and as we add in more images, we end up with a better representation. It's not a perfect representation. That's because what I'm showing you here is a very simple back projection. If you now imagine what's happening in Fourier space, we're effectively adding up planes in Fourier space, but where they overlap, we're massively over-representing our objects. So we apply a weighting which deals with that. That's typically R weighting. So now we generate our projection, we weight it, and we back project the weighted projection. And if we now do this with a large number of images, we end up with something that looks much more like the object we put in. You see that I've illustrated some of these things by going into the kind of Fourier space equivalent. You can think about this as Fourier reconstitution as well, but in practice it's done by back projection. So what software do we use to do this? There are multiple different packages available. Data collection on the microscope, there's Cryo-EM, there's FEI Tomo, there's Leginon. We make use of Cryo-EM for this. For tomogram reconstruction, so for taking the data and generating the reconstruction, there's IMOD, Tom, ProTomo, PyTom, and others. And in the lab, we mostly make use of IMOD. OK, so that's tomography. I'm now going to move on to subtomogram averaging. So let's imagine we've collected a tomogram like this. And this is part of a cell. And you can see in here, for example, you can see lots of ribosomes. These are these black dots here. You can see microtubules here. We have a relatively low resolution three-dimensional reconstruction because it's one lot of dose applied in order to generate this reconstruction. But we can potentially cut out repeating objects from this image and average them together. And if we do that, we obviously improve our signal to noise, and we can interpret the average out to higher resolution than we can the individual images. So the basic approach is extracting volumes which contain the object of interest. They come out in some potentially random orientation. They're aligned to some kind of reference, so they're all in the same orientation. They're averaged together. The average is used as a reference, and that process is repeated. This is very similar to single particle reconstruction, but we're using three-dimensional volumes that are being aligned and averaged rather than two-dimensional projection images. If we look at that as a workflow, we're generating our data on the microscope, the Ctf correction, the tilt series is aligned, the tomogram is reconstructed, the subtomograms are extracted. There are potentially initial Euler angles assigned. If, for example, we're reconstructing a protein which sits on membranes, we can see the membrane in the tomogram. And that means that we know that the object is sitting somewhere in one of these directions. It's not sitting like this next to the membrane. So we can pre-assign some of those Euler angles. That's effectively defining priors. The subtomograms are aligned in 3 dimensions. They may be classified. They're then averaged. And that average is then post-processed in whatever the appropriate way is, and we generate some kind of structure. There are multiple different packages for doing this, and in some cases, there are split packages, so we may use a standard tomography package like IMOD, for example, to reconstruct our tomogram, and then a specific subtomogram averaging package to do the rest of it. Other packages can do the whole process. Widely used packages include Dynamo from Daniel Cassano-Diaz in Basel, PEAT, which is from the same group that develop IMOD and Cryo-EM, PyTom from Friedrich Förster, and Relion from Tanmay and Jurs here. And these may be maximum cross-correlation or maximum likelihood-based approaches for subtomogram averaging. Okay. So why do subtomogram averaging? So one question is, why not just do single particle reconstruction? There are certainly borderline cases. If you imagine you've generated a lamella which is extremely thin, then you can probably just take a 2D projection image, pick out the things which you're interested in, and do single particle reconstruction. at some point your lamella becomes so thick that you can't then find your object of interest anymore, or you can't align your object of interest anymore. So I think at the moment it's a safe thing to say that if you can do single particle reconstruction, then you should do single particle reconstruction because it's much quicker to get large amounts of data. The packages are better established and you'll get to a higher resolution structure much faster. But subtomogram averaging allows structures to be determined when other objects in the path of the electron beam would otherwise prevent alignment. Subtomogram averaging may also have potential for application in single particle type projects. People have used it for generating starting models, and there's also potentially more information in a properly collected tomogram than there is in a single 2D projection image that may help to align certain objects, but that's not a generally used approach. So if you do subtomogram averaging, you get two kinds of information out of the analysis. The first one is hopefully you get the structure. So this is a hexamer which packs together to form the mature capsid core of HIV. So you get your average structure or multiple averaged structures if you've done some kind of classification. But you also get the position and the context. So you've picked each one of those subtomograms out of a volume, you've done alignment, you've worked out the angles and the shifts, which means you know exactly where each contributing subtomogram was in your tomogram. And you can visualize that. So in this case, we've placed one of these hexamers at every position where we found the hexamer. And that gives you the structure of the core, as well as the structure of the component protein, which was actually the target of the subtomogram averaging. And the other thing you get out of subtomogram averaging is the context. So you're not looking at a protein complex that's just floating around in solution. You may be looking at it in something that's much closer to its native environment. So in this case, for example, the relationship between the core and the viral membrane may be something of interest. If we've reconstructed other proteins, we would see their relationships to each other. So we get the contextual information. So subtomogram averaging, as you're aware, remains a relatively niche technique. There are not very many labs that do it, and that's because it's still quite challenging. So why is it challenging, and what are the challenges that we may want to overcome in the future? So the first one is sample flexibility and heterogeneity. It's generally the case that the kind of things we look at by subtomogram averaging are more heterogeneous and more flexible than things we look at by single particle, typically because there's been no attempt to purify the sample. There may have been no attempt to generate a homogeneous construct. We may have all kind of cofactors bound or not bound. We may have various other cellular proteins attached or not attached. So we may have increased sample flexibility and heterogeneity. That's a fundamental problem which we're not going to get over. We have higher apparent sample thickness. Very often the targets of tomography are thicker samples, but if we're tilting the sample, it's getting even thicker. We have two alignments and reconstruction steps. So we generate our tomogram, and then we take all of those tilt images, we align them together, and we average generate our tomogram. We then extract our subtomograms and do another alignment and reconstruction step. So we have two steps rather than one. Sample movement and change during data collection. When you do single particle reconstruction, you generate movies, and you know that your sample moves around a bit, and it may change a bit over time. The same thing happens in tomography. So as we're collecting our tilt images, our sample may be physically changing as we collect the data. That means that our image from this angle and our image from this angle may no longer really be of exactly the same object because it may have changed. We're typically using higher total electron doses. We typically have much smaller data sets because the data collection is much more time consuming, and it can be more difficult to determine and correct the CTF because we've got much lower signal to noise individual images, and we've got a three-dimensional volume challenge. So I'm going to briefly talk in a little bit more detail about two aspects, just to give you an idea of the kind of things that are going on to improve this method. So the first question I'm going to ask is, in which order should we collect our images? So I'm going to start with what's called the continuous tilt scheme. This is quite a widespread approach to collecting the data. So we're first going to collect the 60-degree image. And then we're going to rotate the sample and collect all of the other images. What's illustrated here, so if you imagine these are then this is then Fourier planes that are being added into our reconstruction, and they're being multiplied by a filter which represents the loss of information due to accumulated dose. So as you can see, the first image contains beautiful high-frequency information, and as we go through the tilt series, at some point we've applied already 80 or 100 electrons per square angstrom, and there's no more high-frequency information left. So all of this information is missing, as well as the missing wedge. So we can do a bidirectional tilt scheme. We start at the middle. Perhaps I should just say one thing. The other thing that's happening is you can see that even the low-frequency information is lower at the high tilts than at the low tilts, and that's because the sample here appears thicker. So let's do a bidirectional tilt scheme. We start in the middle, we go in one direction out to the end, we go back to the beginning, and we go out to the end. This has advantages because the high frequency information is collected first, the low tilts are collected first, the high frequency information is preserved, and these are the images where we've got the thinnest sample. So we're collecting the high frequency information when the sample is thin and not when it appears thick at high tilt. But we're going out in one direction and going back and then going out in the other direction. And the various distortions have accumulated in the sample due to exposure in this time. And when we jump back, the sample image at this angle and at this angle may be different to one another. And you can actually see that. I'll come back to it. Or we can do a dose-symmetric tilt scheme, where we start in the middle, and we just oscillate up until we reach the edge. And that, as you can see, gives a symmetric distribution of dose. So that's a summary of the three: continuous, bidirectional, and dose-symmetric. And if we consider the effects of the accumulated dose and the thickness of the sample, we can look at the mean signal transfer over resolution, and it's different for the different tilt schemes. So the continuous is worse, but for the dose-symmetric and the bidirectional tilt series, we get better signal recovery. What this plot shows is an estimate as to, as you go through neighboring images in the tilt series, how much difference there is between the two, and that's based on looking at the movements of the gold beads. And in black is the dose-symmetric tilt scheme, and in green is the bidirectional tilt scheme. And this big jump here is the change when you go from here to here. So when you come back, and that's the movement due to the accumulated dose. And in the dose-symmetric tilt scheme, that's distributed at these high tilts where we're not interpreting high-resolution information, so it's much less damaging to our reconstruction. So how we collect the data can influence the quality of our reconstruction. The next thing I briefly want to talk about is Ctf correction. So if you imagine three objects, a red one, a green one, and a blue one in our sample, and we image them at 0 degrees, and we image them at 60 degrees, in practice they're at different heights within the sample, and they're therefore at different defoci. So if we simply correct all of our images using the average defocus, we'll be using just this defocus, and we'll be underestimating it for one and overestimating it for the other. And those of you that do single particle will know that the effects of messing up your CTF measurements are very severe in terms of the quality of your structure. What's most commonly done is to take into account the fact that when you tilt your image, one side of it is at a different defocus to the other side. And nevertheless, one would then use a single defocus value to do the correction. So if we imagine we use defocus value D to correct the 0 tilt image, then this would be compensated according to the tilt, and this would be compensated according to the tilt. So we take into account the gradient of defocus across the slab due to the tilt. But that's not the only problem. We're working with thick samples, and therefore we also want to take into account the defocus gradient through the sample. So the red bead is not at the same defocus as the green bead. So this is the effect of reconstructing beads this is what the reference image looks like. So this is a simulation based on a tomogram where there is no CTF. This is what happens if you reconstruct your image without doing any CTF correction. You can then see the effects of the CTF on your bead. This is the effect of doing the reconstruction using the traditional two-dimensional CTF, where we take into account the tilt of the sample, but we don't take into account the depth of the sample. And you can see that beads at different positions all look much more like reality than if we don't correct, but they all look different. And this simulation here is the effect of doing a true three-dimensional CTF. And this just gives you an idea as to the effect of that. If we do a 2D CTF versus a 3D CTF, we get an improved Fourier-Shell correlation, and we can interpret higher resolution information. So to come back to our challenges, sample flexibility and heterogeneity is a fundamental problem, which if we want to look at objects in cells, we have to embrace rather than try and fix. It becomes the heterogeneity and the flexibility that's actually interesting. The higher apparent sample thickness can potentially be compensated by sample preparation methods that allow us to prepare thinner samples. It can be compensated by collecting the tilted images late in the series when we're only pulling in low resolution information anyway from those. So we can partially offset this problem. There are approaches for combining the reconstruction of the tomogram and the reconstruction of the final subtomogram average into one reconstruction step. And this will continue to develop as the computational packages develop. The sample movement or change during data collection, we're never going to get rid of it completely, but we can, again, mitigate the problem by collecting the high resolution information early in the low tilt images and placing the distortions out at the high tilts where we've only got low frequency information. The higher total electron dose, in the end we can set our electron dose at whatever level we want. It's just a matter of what we actually need to get alignment. We have much smaller data sets due to more time-consuming data collection, and this again is something that can be optimized. We will get faster and faster. I don't think we're in the foreseeable future going to be as fast as single particle, where you can just collect a whole series of 2D images, but there's clearly room to optimize the amount of data that's collected. And there are already improvements in how to correct the CTF. So many of these challenges will be overcome, and subtomogram averaging will become an easier to apply method and a more routinely high resolution method in the future. So tomography has traditionally been a low resolution approach. Subtomogram averaging can go to high resolution. This is a structure that we did last year of part of the capsid shell of HIV. We now have this structure at 3.4 angstrom using tomography methods. So it's very rare that there's a fundamental problem or a kind of a theoretical difficulty in getting to high resolution. We certainly can get to high resolution if all of the steps are optimized. What makes this sample go to high resolution is that it's possible to collect very large data sets because there's extremely large numbers of copies of the object of interest within a single tomogram. And then these illustrate some of the reasons why you might want to think about this. So one is that you get the structure in situ. If you solve your structure in the cell, there's a kind of sense of finality about it. The question as to whether making that construct, cutting off that domain, running that purification protocol, what that might have done to your sample, those kind of go out the window if you solve the structure directly within its functional context. You get the cellular context. So you see your structure in its environment where you can look at all of the other things that are around it and how those might be influencing the biology that you're interested in. There's the potential for structure for discovery, by which I mean that you can, for example, you want to solve the structure of the ribosome in the cell. You can go in and solve the structure of the ribosome in the cell and apply classification methods and look and see what cofactors happen to be bound to different ribosomes at different places in the cell. So we can potentially discover conformations and cofactors that we may not have necessarily known about simply by going in and looking. And I think in the longer term, there may also be potential single particle applications for particular cases. It's a little bit embarrassing to show only one reference and it be one of your own for the end of the talk. But I just realized that the review's very recent, and we cited an awful lot of very good papers and very good reviews by other people in here. So rather than write up a long reference list, look at the reference list for that in that review, and you'll find lots of things that will hopefully tell you more about tomography and subtomogram averaging. Okay.